Thank you very much for the invitation to be here as well. It's great to come back to campus. I really enjoyed walking and talking around and meeting with people. Brought back a lot of memories, even though it's been a few years, right? I won't get into how many. It's also really cool to give this seminar. I can remember when I was a student 10 plus years ago and looking forward to the monthly neuroprosthetic seminars, and it was really cool to see external speakers. So it's nice kind of full circle coming back and giving a talk. So yeah, I'm going to focus today on one of the primary areas of our research, working towards bladder control, and there's some other avenues we investigate as well. So it's something for those of you enjoying some of the beverages provided outside. Hopefully you can maintain your bladder control for at least another hour or so, and then make a beeline for the restrooms. Right at the beginning, I want to at least acknowledge some initial funding also. So so Nielsen Foundation gave some initial grants to help, one initial grant to help the lab get started. A number of these experiments were done under some NIH funding. I'll mention some stuff at the end that's supported through an NSF award, and then Medtronic has been supporting some of the closed loop work that I'll touch on in the middle later parts. I also want to give a shout out to my home department institution. So if there's any undergraduates thinking about graduate school at another place or grad students thinking about an external postdoc, we've got a really strong neural engineering program at Michigan across different areas. We have a pretty good neural engineering community and BME community, but neural engineering as well across different disciplines. And there's a number of us who trained here in Cleveland as well, got good foundations. So my lab is the Peripheral Neural Engineering and Neurodynamics Lab, or the P-Neuro Lab. And we kind of fall under this realm of kind of a title called bioelectronic medicine that's growing in use in the last few years, in recent years, of innervating or controlling different autonomic organs to restore function in different ways. So that's one of our primary goals, is to look at and investigate organ control. So bladder function being one thing, carrying on from some of the prior work I did in my PhD, ranging from overactive bladder and incontinence, where people have challenges keeping urine in their bladder, to detrusor underactivity, where individuals have an ineffective bladder emptying. We've also branched out looking at neuromodulation for sexual dysfunction, done some studies investigating renal nerve stimulation for potential diabetes applications, and we also do a lot of work on collaborating on the development of novel interfaces, including some work looking at new electrodes designed for sacral roots and dorsal root ganglia, and developing applications on some small microneedles for autonomic nerves, and right now testing out in the vagus nerve, although there's other applications. So today I'm going to focus mainly on our work on bladder control, and later towards the end I'll highlight some of the new electrodes our labs are working on that may have applications in improving or pushing along this therapy. So as I imagine many of you may be aware, or perhaps you're not, there is a significant population in the US and worldwide that has bladder problems, at least 30 million individuals, give or take. And we often focus on individuals with spinal cord injury who lose bladder function, being at the tail end of their spinal cord after an injury. And among those participants, there's surveys from Kim Anderson years ago, and one at the University of Pittsburgh I was involved in with Jen Collinger, where we asked, and spinal cord injured patients often report that bladder, as well as bowel function, is one of their most important priorities. There's also a large population group with non-neurogenic bladder, where, say, after childbirth, aging, and other effects, they can have incontinence or overactive bladder symptoms, or sometimes detrusor underactivity. So for these individuals who may go through primary lines of treatment, such as medications or behavioral changes, a neurostimulator of some sort is often the third line of treatment. And so for the non-neurogenic patients, so those usually without problems in their nervous system, a sacral nerve stimulator has long been a standard of care. And so Medtronic has an InterStim device where a standard stimulation lead is placed through a hole in the sacrum to target a sacral nerve for a long-term implant. There's over 200,000 of these implants. And more recently, there's a couple of competitors coming into the market. Axonics recently started pushing through in the United States as well, and go from, say, an InterStim device, which has a battery that gets changed every 4 or 5 years or so, to Axonics, which is rechargeable and has a much longer potential battery life, although individuals then have to make sure and recharge their batteries. Another therapy is this PTNS, or tibial nerve stimulation, where patients may come into a clinic and they have a needle placed by their tibial nerve at their ankle for a 30-minute session once a week for about a few months, and then they have maintenance sessions once or twice a week thereafter. So a very different type of therapy that does, I think of it kind of like a physical therapy for the nerves, even though, of course, and it somehow affects the bladder function, which all of us are still trying to figure out. In both of these particular cases, neither of these stimulation paradigms are necessarily performed with respect to the specific bladder symptoms. So it's always on in one case or periodically being used, being applied. And then for spinal cord injured patients, the Brindley approach or Vocare approach, as was used here in the US for a while, can be used to directly stimulate the bladder to help empty it out. For the primary therapies used for broader non-neurogenic patients, this open loop stimulation is continuous and isn't based on bladder needs. So a goal in our lab is, can we do more of a closed loop stimulation? Can we inform the stimulation timing and potentially improve therapy?
Can you comment on the relative success of these strategies, particularly the percentage respondents?
Yeah, so broadly speaking, it's about 2/3 of people get the clinical target of about 50% improvement, who are considered for the therapy and then get the long-term implant. So say, to be more specific about the InterStim device, it's a two-stage process where first you get a temporary lead, and if you have successful responses after a couple of weeks, you get a permanent lead. And about, 50-60% of the people respond to the permanent lead. I'm flipping those numbers, but it's about 2/3 and 3/4 at each stage. And you end up about half to 2/3 get long-term benefits. So there's an opportunity there for improved therapy, perhaps with a more selective interface. And it's fairly similar to PTNS, but the nice thing about PTNS is it's much easier to either start or stop that therapy. And along those lines, there are a drawback is that right now it's done in the clinic. And so you can imagine perhaps not wanting to drive into the hospital or somewhere to do this on a weekly basis. But there are some groups that are investigating implanted stimulators to leave in somebody's lower leg to do kind of on-demand stimulation. So I'm going to briefly highlight some of the anatomy here as well. And so this is a very simplified schematic of the bladder and lower urinary tract. And so here we have bladder as the storage vessel for urine, urethra as the outlet path, and the urethral sphincter as a closure mechanism. And two of the primary nerves are shown here. So the pudendal nerve innervates the sphincter, urethra, and other parts of the pelvic floor. Provides for us our somatic innervation, where voluntary control over the sphincter and sensations coming through it. Pelvic nerve provides parasympathetic innervation of the bladder. So here it's kind of that contraction of the bladder itself. We also sense bladder fullness. So when we start to feel our bladder being full, it's generally through the pelvic nerve. Not shown here, but I'll touch on briefly later. There's also the hypogastric nerve, which comes from a higher spinal level, and that gives more of the sympathetic innervation, so to maintain continence in the bladder. And back before I started graduate school, Ken and some colleagues and others had showed that if you stimulate the pudendal nerve, generally around 20 to 33 hertz, or within the urethra in some human patients, you can evoke a kind of a micturition or a bladder excitation response. So micturition being a fancy word for emptying your bladder. And so in my PhD, I investigated this a little bit further with 3 studies. So the first study was putting cuffs on the pudendal nerve itself and investigated the use of non-continuous stimulation patterns. And so, typical electrical stimulation oftentimes is performed in this kind of monotonic, constant approach. But using a seed idea from Narendra Bhadra, I looked at using bursts of stimulus patterns and ended up seeing 20 to 30% larger bladder contractions by using these kind of alternate stimulation patterns. Next, I placed catheter within the urethra and looked at mapping out the pathways a little bit. And by stimulating at different locations in the urethra, in both intact and then spinal transected animals, observed that there was at least 2 pathways, as different locations in the urethra responded to either a low frequency at 2 hertz or a higher frequency at 33 hertz differently. And then in a third study, we placed some fine wire electrodes, actually EMG needles, into the urethra at different locations. And when I used a bursting stimulus at locations closer to the bladder neck, was able to get functional bladder emptying. In this case, functional meant something on the end of 80% of the bladder was empty out, or 80% efficiency, although it was across a matter of minutes. So this was a rather slow void, even though it was functional. So I don't know if it's a little depressing to see an entire PhD summarized in one slide, but But this is all done in anesthetized animals as well. So there's things that we can learn, but it still is not quite the same as awake behaving animal model. And a question here as well is, can we inform this stimulation based on the bladder state? So in each case here is trying to drive stimulation at different times. And so some of the forward-going work then is flipped more on maintaining urine in the bladder, so the continent side of bladder function. So returning back to this diagram, as I mentioned, stimulating on the pudendal nerve can drive a micturition or bladder contraction reflex, stimulating around 20 to 33 hertz and some lower frequencies. It's also been shown and continued research on if you stimulate at around 5 to 10 hertz or so at the pudendal nerve or some of its branches, can drive a continence reflex through the hypogastric pathways. And as I mentioned, you can observe the bladder state through the pelvic nerve. However, while the pudendal nerve can be accessed different ways in humans, some of the branches are very superficial. The pelvic nerve is a deep plexus. It's nothing like this simple line here drawn on the diagram on the board. And so in our animal model and in humans, it's kind of like a spider web. And so there are not good locations to put one individual electrode on, if you want to say record from the bladder state. However, both the pudendal and the pelvic nerve converge at the same spinal roots. In our cat model, it's S1 and S2, rats and S3, rats is L6 and S1, humans, it's S2 to S4. And the dorsal ganglia are locations where the cell bodies for these sensory fibers are placed together. And so the general concept we had was placing a microelectrode at that location. We could perhaps selectively stimulate individual pudendal pathways to drive function and record from individual pelvic fat pathways to identify the bladder state towards a closed loop interface. And so I got down this pathway, at least moved toward this pathway, in working with Doug Weber in his lab at the University of Pittsburgh while I was a postdoc. And so between Doug and myself, and then also Rob Gaunt, we did a number of experiments looking at sacral level electrodes for monitoring bladder function. So this is the basic setup from then, and in many of these experiments, it's what's being used in my lab in continuing this work, where electrodes that typically use are these black rock electrode arrays. So it's similar type of electrodes used in brain-computer interfaces, except put at a peripheral nerve location. A lot of benefits in working with them. They're pretty robust. In acute experiments, we're using them over and over again. And we're able to insert them through the epineurium of the DRG into the tissue pretty well. So this is an example of one of our recordings. In fact, back from a paper during my postdoc at Pitt, where we may see the bladder pressure in the black line going across. And so there's two larger contractions and some smaller bladder, smaller waves along there. And then what we record from individual channels would be a continuous voltage stream. And whenever the voltage crossed some threshold, we'd grab that as an action potential occurring, or we call it snippet. So examples of snippets are on the far right for the bottom channel here. And each of those hash marks, each of those lines, what we call a raster plot, is the occurrence of an action potential. So when going across, when the bladder pressure is relatively flat, the action potential rate is more or less constant. And during a bladder contraction, there's an increase in the density of the action potential spike rate. So it gives us potential information for turning into an estimate of the bladder pressure. Continuing in my lab at Michigan, first in experiments led by former postdoc Shani Ross, we would map out and see where we would see bladder neurons. And so here's some top-down views of electrode array in different dorsal root ganglia, either on the left or the right side at S1 and S2, so different levels. And across experiments, there'd be a lot of variability in where we'd see these signals. And basically what we're doing is putting 30 to 50 channels to a place where there's thousands of axons passing through, and we're sampling from some subset of them. And during these experiments, we observed that it's not a clear linear relationship between the smooth firing rate of these neurons and the bladder pressure itself. So there's some hysteresis between them. As example here, at the bottom is a bladder neuron, and it hits its max firing rate just before the bladder pressure reaches a peak and returns back to baseline while the bladder pressure is still relaxing. This hysteresis is not surprising necessarily, as the bladder muscle is known to have a nonlinear stress-strain relationship, as plotted down below. And so across a series of experiments and observing bladder neurons, we look to quantify this by looking at the range of, we call hysteresis in this. So comparing the firing rate range across a range of bladder pressures. And across these experiments, there was consistently saw that all bladder neurons had some amount of hysteresis. The specific numbers and ranges aren't too critical there, but the fact that it's at least a moderate level of hysteresis, so we couldn't turn just a simple y equals mx plus b, what's the firing rate, multiplied to come up with a more accurate bladder pressure estimate over time. And so in continuing forward, Shani and PhD student Eileen Ouyang were looking at different algorithms then to turn this spike train into a reconstruction of the bladder pressure. And in a paper that we had come out last year, they looked at a combination of different decoding algorithm components and looking at different components into getting a improved estimate of bladder pressure. So at the left, we would take signals from the dorsal root ganglia, and in this case, just taking thresholded activity. So if you've heard about spike sorting, in this case, we were not sorting out into individual units, but just saying how often any channel had a rise in signal correlating to some underlying action potential activity. So we would consider maybe different firing rate intervals, or how often we would be counting spikes in different bins, ranging from 100 microseconds up to 2 seconds, whether this firing rate was smoothed in some fashion, normalized, so firing rate normalization, different approaches for selecting channels into the model, either all channels or some subset, and different types of algorithms that we also considered. And we did this across 5 new anesthetized experiments where there were different bladder fills, where we would test the model or develop a model, and then a separate bladder fill where we would test that. And all of these particular analyses were done offline or after the fact. So right off the bat, we noticed that the smoothing and the normalization were not important in our overall model development. In considering the firing rate calculation interval, here's an example from one experiment that was pretty consistent across others that we saw, where there's 2 columns here, or 2 rows here of a array at the top on the ganglia, and we're considering different intervals or bins at which we're counting spike rates, from either a short bin on the left or a longer bin on the right. And in the short bins, we would have a lot more data for the model, basically more time points we're entering in to try and estimate bladder pressure. But on the right, we had ability to get much more smooth rates and got more correlated channels. And this is ultimately a better use for our model, as the bladder is a kind of a slower moving physiological parameter than, say, if you're trying to decode finger function or something along those lines. For channel selection, we looked at different types of approaches as well. When we considered using all channels within our model, we would see that occasionally unrelated or spurious channels would have random artifacts that could really affect our outcomes. And so we narrowed down our interests in only channels that had correlated signals. And so of course that kind of makes sense. If we only took the channels that had a high correlation to bladder pressure, of course we're going to get a better output. But the challenge was sometimes if we set a really high threshold, then we may only have a few channels. And so we would work in a stepwise function to kind of identify our highest channel correlation coefficient to bladder pressure that yielded multiple channels. And then finally, we would consider a regular linear regression, a Kalman filter, which is commonly used in brain-computer interface applications, so more advanced linear algorithm, but with different types of smoothing incorporated, and then also a neural network with one hidden layer. And so this is an output of one common type of, or one experimental analysis output, where we might have the bladder pressure in black, and then in this case we used 5 channels, and then just fit the firing rate for those 5 channels with the algorithms to come up with an estimate using either the Kalman filter in red or the linear regression in blue, and a decent overall fits to the bladder pressure. And then on a subsequent trial within the same experiment, we'd take the same model, apply it to the firing rates, and come up with a pretty reasonable estimates of bladder pressure in this case. So an normalized error of around 9 to 10%. So across the 12 data sets that we worked with, we saw some variability in the results and some trends as well as far as which algorithm worked better. When we were considering the normalized root mean square error, the neural network model had the most consistently had the lowest normalized error. And so here we see it's potentially an algorithm to use when you're looking to set a certain threshold. If the bladder hits 20 centimeters of water, for example, we need to do something. And that may have the best chance of having good response rate. When we looked at a correlation coefficient, as far as following the overall trends, the Kalman filter did a lot better on that end. And so it had a consistently higher Kalman filter output, or the higher correlation coefficient. So here, this is our ultimate interest, was if we're looking to detect bladder trends, is there kind of a rise in the pressure, even if maybe our offset or our baseline value might be slightly shifted. So in continuing that work, then Eileen has done an additional set of studies that we had published earlier this year in IEEE, TNSRE. We're trying to do kind of a real-time decoding. So the prior work was we'd collect neural signals, and then we'd go back and kind of analyze them offline to see what algorithms work the best. And in this case then was taking what we identified as sort of the Kalman filter and some of the parameters and doing real-time state decoding. Additionally, we did many of these fills with trying to simulate an overactive bladder scenario. And so we filled the bladder with an acetic acid, which is an irritant, and so it would cause the bladder to contract earlier and have a smaller bladder capacity. So simulating a overactive bladder. So in this multi-channel or multi-plot diagram up at top here, first we would do a training run where we'd fill the bladder and record and see which channels correlated to the bladder pressure. And so by the one indicator there's two 4x10 arrays that were in two different ganglia, and by color scheme, the darker red ones had a higher correlation. And then a little bit later was this other larger plot of the bladder pressure. And then there was applying the Kalman filter in real time and watching see how well it did. In this case, it followed the overall trends pretty well. And the individual smaller contractions before the large rise were also tracked, although not to an exact value. And then at bottom shows examples of some of the threshold crossings that we were observing, although in this case they ended up being very large single unit action potentials. And this is an example where in some cases there were neurons firing early on during the bladder rise, and you can see the on and off period of their action potentials that correlated really well with when bladder pressure oscillations, and others that turned on much later during the more large contraction at the end. And so here we're using a microelectrode interface, getting multiple different perspectives on the bladder contraction. So across, and this is a new set, these 5 new experiments, looking at both saline bladder fills, so kind of a control fill, as well as the acetic acid stressor fills, we got overall, on average, a really good correlation coefficient. So 0.84 for saline and a higher correlation coefficient, 0.95 with the acetic acid. So it's kind of interesting to see we got a better fit in the, when we were using the overactive bladder, probably an increased sensitivity in those cases. One thing that was interesting though, is that we would observe across like one experiment, and this would be running for 10, 15, 20 hours, we would see signals that would go in and out on individual channels. And so the correlation between these channels' firing rate and the bladder pressure could vary during an experiment. And so we wanted to see if we would have been better served by sorting out, by looking at individual action potentials in those trials. So starting here is plots for 3 single units, so 3 individual action potentials in one experiment across different trials, 1 through 9. And their correlation coefficient to bladder pressures is plotted. And there's a lot of variance in this one individual experiment, as well as across other experiments that we could find individual single units that persisted across the entire trial. And in comparing this to threshold crossings from the same experiments and kind of selecting individual ones, we see about the same amount of variability, whether we were just selecting thresholds for individual channels or trying to sort out individual units. And so at least in these acute non-survival experiments, we got about the same amount of variability, but we saw many more of these threshold crossing channels than single units. I mean, there was one experiment, experiment 2, where there were no single units that we could clearly identify, but plenty of threshold crossings. And I'll touch touch base later on as to why we may have been seeing this in and out of signal going on. It was likely little bit of a settling of the electrode within the ganglia across the short-term experiments. So now that we could at least have a reasonable estimated bladder pressure during these non-survival experiments, moved on, can we do stimulation as well? Can we get selective stimulation of pathways? So starting from some prior work at Pittsburgh again, we did some experiments where we were doing microstimulation to evoke responses. So in the map at top right there's an array on top of the ganglia, and individual channels are stimulated one at a time for 5 seconds, as shown in the middle plot. And then the color coding just indicates which ones have larger or smaller contractions. So here's examples of some large contractions on channels close to others that did not evoke a response. So some selective activation of pathways. We could also selectively or unselectively activate relaxation pathways, either with many channels stimulated at once, so all the blue ones lit up, leading to bladder relaxation during the gray period when stimulation is on, or individual channels being stimulated for brief periods of time, showing some start of bladder pressure dropping there. We continued and did some similar sorts of evaluating of parameters in the lab at Michigan. So here's an example of stimulation is turned on in the gray period, and the bladder pressure in blue drops off during stimulation. And at the same time, we're recording from neurons, and here's a particular bladder neuron whose firing rate correlates very well with bladder pressure. So there's the potential for detecting bladder state with that recorded neuron and also stimulating the control function. So then worked on try to do some closed loop trials. So here's, I think, probably our first closed loop trial, where it was using in this case, a neural network algorithm. 10 hours later, after the model was fit, did another bladder pressure trial and did real-time estimation of the bladder pressure. And so the pressures in blue, the estimated pressure in green follows the overall trends, okay? And then once it hit a kind of arbitrary threshold, we turn on stimulation automatically. And so here, just 4 channels of DRG led to quick bladder relaxation. But at that point, we were not decoding during the stimulation as well. And so each time stimulation was cycled on and off. But at least it showed potential for doing closed loop stimulation. So then Eileen updated this overall algorithm and looked at integrating some artifact rejection schemes. And so here's another closed loop trial it did more recently. And instead of doing in dorsal root ganglia stimulation, shifted out to doing pudendal nerve stimulation. So one challenge we had in working with DRG stimulation was there's many channels to work with, and it would take quite a while to try to characterize every channel and figure out, well, which ones are excitatory, which ones are relaxation. And so for the purpose of some of these experiments, it was easier to just move the pudendal nerve and do some additional stimulation point right there. And so here's an example of a closed loop trial where initially the decode has a little bit of an offset, but once the bladder starts oscillating in the middle with the bladder pressure, the decode follows it pretty well and turns on for stimulation. And so in 2 closed loop trials here, we had an average bladder volume of about 31 mLs. Without stimulation, the average bladder volume was around 22, 23 mLs. And so we had about a 40 to 50% increase in bladder volume in these couple of initial trials. So this approach showed some promise for doing closed loop stimulation. So our next interest, we're moving to maybe more of a clinical related stimulation approach. And so through a grant with Medtronic, we're using their real-time developer kit to do some closed-loop neuromodulation of the sacral nerve, so back to their sacral nerve stimulation approach. And so this RTK, or this Summit API system, is something that they developed for some adaptive deep brain stimulation. And so then we're also applying the same technology and tools to trying this closed-loop bladder control. And so a similar type of setup of microelectrodes in dorsal root ganglia, and then triggering algorithms to drive their INS, or implantable neurostimulator. And so the ideal plan was to use their clinically used stimulation lead in our animal experiments. However, the animal nerves are much smaller than human nerves, and so there's too much current spread in our experiments. So we moved to using a nerve cuff that we then just connect to the stimulator. And first we did some work to quantify, or at least analyze the primary parameters. And we saw for doing sacral neuromodulation, about a 5 Hz stimulation had the best effect for relaxing the bladder at about twice the motor threshold. So that would be the amplitude of stimulation that was twice the level where we saw some twitches in the perineal floor. And for algorithm detection, we would see about a 6 centimeter increase in water. So we saw increase in 6 centimeters in about 4 seconds was our best detection algorithm for observing contractions. So even here, there might be some offsets using that Kalman filter, but it still would follow the overall trends pretty well. So here's an example of what one of these closed loop experiments would look like, where the bladder is being filled from left to right constantly, and once eventually there was some bladder leakage, we call that our capacity. And in this case, stimulation was turned on when detected a contraction. So even here, although each small contraction was not detected, we were still at least applying stimulation in many of the epochs. So the goal here then was to compare 3 different types of trials, either those with no stimulation, so it's like a control fill, continuous stimulation as is done clinically, or this closed loop approach. And across a series of experiments and trials, we saw about a 15% increase in the bladder capacity in using these closed loop stimulation paradigms. And so this was, for individual trials, this was even a higher bladder capacity than doing continuous stimulation, but only using stimulation for about 40% of the time. And it was interesting to observe that the bladder fill, when the stimulation was applied, kind of varied across the bladder fill. So the x-axis is looking at different percentile or quartiles of the overall time of filling the bladder itself. So the first 25% or the last 25%. And about half of all stimulation is applied in the last period, which would be expected. But some early stimulation also applied probably helped maintain or drive continence a little bit further. And yeah, I guess I'll briefly comment as well. So I was thinking about this, we're hoping for, anticipating maybe a larger increase in bladder pressure than the 14 or 15% seen here. But this number, at least the output we got, was consistent with trials or experiments that have been done elsewhere using the same sort of anesthetized prep and allowing some of the other setup that we had used. So that was positive to see. And so while this shows promise in anesthetized experiments, the next goal is, can we do this in awake and behaving animals? I mean, that's ultimately the kind of important translational step. And we've done aspects of that in different directions. And so one component is we did some awake stimulation last summer. And so this was part of a subcontract we had with Margot Damiser and Dennis Barbeau, and providing algorithms to their work with Rob Karam, where they're developing their bladder sensor. And so we were doing some awake behaving recording of cat bladder pressure. And in 2 animals, we put a cuff in on a nerve cuff on the pudendal nerve. And so while filling the bladder through catheter and monitoring the pressure, we'd either sometimes no stimulation, sometimes apply 5 hertz stimulation to perhaps promote continence, and other times apply 33 hertz stimulation, which may promote bladder emptying. And so this is an example of one of the experimental recording sessions where bladder pressure across time, and after an initial large peak in the bladder pressure, we're letting the bladder system kind of stabilize. We'd either apply no stimulation, or then 5 hertz in the kind of yellowish color, nothing, and then 33 hertz in the reddish color. And you may observe in here, so there's what we'd see during our acute experiments would be kind of a nice flat baseline until something starts happening. Here, there's jumps up and down, some artifacts, and there's the animals moving around. It's kind of more of a real world scenario, to the extent that you can herd a cat in a little cage area. And kind of the primary parameters we look at is either the interval between bladder empties, so it may be related to maintaining continence, or the efficiency of when a voiding event occurred, so relating to how good the micturition event was. And so across these range of experiments, we had a lot more no stimulation trials, partially because that's kind of the primary objective why we were doing these trials, but also because we wanted to give some breaks in between stimulation to try and let the bladder reset. There was a lot of variability across these bladder fills, which I'll comment on. But we noticed that in general, 5 hertz at a higher amplitude led to a longer interval, even longer than at the voiding without stimulation. And also under the voiding efficiency, we observed that the 33 hertz at a higher amplitude overall had a greater voiding efficiency. So a couple of key things here, one being as seen under anesthetized experiments, 5 hertz or the lower frequency was better for maintaining continence, and higher frequency was better for emptying the bladder, and also that higher stimulation amplitudes led to a better response. And this is a case where the animals tolerated this twice motor threshold very well. I mean, when it's turned on initially, a minimal response, if any. Higher amplitudes, they started to act like they could feel what was going on, so we could tell it was starting to become uncomfortable. But the challenge is we're trying to activate sensory pathways, and when you stimulate a nerve, first you're going to get the motor pathways, depending on the type of nerve and everything, but you need to increase the amplitude in this case to get sensory pathways. And so it could be questions on if we got even higher amplitudes, we might have had an even stronger effect, but the animals may not have tolerated it as well. To come back to the variability here, definitely part of this was in working with behaving animals, walking around, moving. They were different moods on different days, or which undergraduate student was helping with the project. But also we also had a couple of catheters in the bladder that certainly caused some irritation, and so led to some smaller bladder volumes as well. So with potential for awake behaving neuromodulation, also the question of can we do long-term bladder state decoding? And so several years ago, we placed some microelectrodes in dorsal ganglia for a number of months, and this is an analysis pulled together by former student Abid Karam, with a lot of other people in the lab helping and working on it. And we did observe that we can see bladder units, and we can turn them into a reasonable estimate of bladder pressure and decode over time. So here's cherry picking some really good data where at 34 days after implant and 2 weeks later at 48 days, using an algorithm, got a pretty good fit. But in general, we'd see there was a decrease in the number of bladder units or bladder-related activity over time. And so most of the time, we'd lose it after a month or so, we wouldn't see any bladder activity. Except for that green triangle experiment, which was really good, which was awesome to see. But even then, when we're getting 10 to 15 units, there's quite a variability from one test session to the next. And certainly a big factor in the loss in signals over time was that we were using a rigid penetrating array being inserted into a peripheral nerve. And that was kind of expected in different ways. And so sometimes we do an explant procedure and observe that the array kind of extruded itself from the nerve. And at right is a tissue stain using H&E, and each of the red dots is near the tip of the electrode. And around 3 of them on the right and bottom, there's a lot of scar tissue around each location. So it's just kind of an inflammation response or long-term response. And I've circled some of the cell bodies that are not too far away from one of the electrodes, but still there's not much that's close to the electrodes at that point. So even though these electrodes worked very well for being able to do experiments to show the potential for approach, really we need electrodes that are designed specifically for dorsal ganglia. On one hand, to limit potential tissue response like this, but also to mitigate against micromotion, because with signals moving in and out of this, I'm sure the array is moving slightly. This goes back to how we were seeing variability in our experiments, our results during acute experiments. It's likely some settling of the array and some settling of the neural tissue. And so there's a couple of, in planning towards doing electrode development, one thing we're also working on is understanding the underlying anatomy as well. And it's led by former undergraduate Anastasia Ostrowski and recently defended PhD student Zach Sperry, where first they looked at distribution of cell bodies in dorsal ganglia across our cat studies. So here at the end of a terminal procedure, we would remove some dorsal ganglia and do a midsection slice, and then manually circle and identify all the cell bodies. So the image at right has half of them identified at the right. And then turn that into a kind of a normalized distribution of cell bodies so that we can normalize across our different histological slices, as no one DRG had the same similar shape. Can't put like a square on top of it, for example. And then Zach came up with a partition model, which was a statistical distribution of which regions had a significantly higher concentration than others. So in this case, it may be hard to see, but the numbers 1, 2, and then 3 are in the top groups at top, or the sections at top where there is a much higher density. So in general, it's kind of known that cell bodies tend to be distributed more around the periphery, just based on developmental biology. As the axons kind of push through, they push the cell bodies out to the side in this location, but it hadn't been quantified before. So we took these algorithms and these processes, and we are collaborating with Scott Lemke and his PhD student, Bobby Graham, to look at the distribution of cell bodies across human tissue. And so there's the NDRI, is a National Disease Research Interchange. I believe I got that right, but NDRI, you can get DRG tissue from them, and then we're tasting slices and analyzing the distribution of cell bodies more in a 3-dimensional space. And whereas before we did manually identifying every cell body, undergraduate Nicholas Peck-Dimmitt in the lab came up with a GUI to do more of an automated or semi-automated process. And from that, we came up with some partition models for cell distribution. So there's a top one is the cell bodies, and the bottom one is on axons. And as before, it kind of goes from left to right in the color bar here, where there's a greater density here and a lower density at right. And then regions that share a color are not different. And so the dark red region at the central top has a significantly greater density than, say, the solid green region to the right. So here again, DRG cell bodies are more central, a little more superficial. And as would be expected, axons are more central, although we saw slight trends between different axon sizes. And so this work is in review. So taking this awareness of, okay, cell bodies are a little more superficial, Maybe we had concept of doing a DRG surface electrode array. And this was inspired by some prior work at Pitt when I was working with Doug Weber, Rob Gaunt, where we designed an electrode working with Yushik Yoon, John Seymour, and his student Kyung-Won Na. That was a thin, flexible microelectrode that we could place on the surface of ganglia. And with that, be able to observe and pick up single unit or neural activity. So here's an example of, we're excited to see that in some experiments we observed a bladder units. So here's the smooth firing rate in red of a bladder unit, tracking the bladder pressure in gray. And in general, the signals were fairly small, or relatively speaking, a small signal noise amplitude. But that's not surprising, as the epineurium is somewhere between 50 and 100 microns thick. So right there is a layer that we're recording through. But even using this surface electrode, we could do some relatively simple surface mapping of the DRG surface. So here's some placements of different arrays. On the stem part shows the type of tests that were done: bladder, filling, brushing the perineal region, or brushing the tail. And we could indicate regions where we saw different types of signals. So it shows potential that maybe a surface electrode like this might reduce a tissue response if it adheres in place on top of the nerve. However, the big question, of course, if we can get it to stay in place. So we've got some ideas where we were thinking along how we would maintain electrodes in place, but during these acute experiments, it was challenging to maintain electrode location. Also during that same program, or same grant through the NIH Spark program, we also investigated developing a penetrating electrode to sample across the dorsal ganglia. And here, like a typical Utah array has a single electrode at the tip. There are Michigan probes, which have many sites along a silicon shank. However, usually the Michigan probes, they can be challenging to consistently insert through the tough epineurium of dorsal ganglia, as opposed to, say, going through the cortex after you remove the dura. So here, John and Kyung-Won led development of a shuttle that was a robust shuttle with a T-beam shape that we had adhered a flexible array to for delivery. And the concept was insert the shuttle in the array and then remove the shuttle. Although for the acute experiments, we just left the two glued together to see if we can get signals. And so across these, again, we were able to see bladder units. In fact, we saw a lot more bladder units when sampling across the array. So here's one example of the firing rate of one unit where it's just tracking some bolus infusions of saline. That's why it's kind of a weird-looking pressure trace in black. But it saw some nice activity. Additionally, the spacing between the electrode locations was 40 microns. So the cell bodies in dorsal ganglia can be anywhere from 10 or 20 microns up to 100 microns in size. And so it's possible to see individual action potentials on multiple different electrode sites. And so Zach worked with James June, who with his colleagues at the Flatiron Institute have a iron-class spike sorting software that allowed us to do some additional analyses and looking at it. So in this complex plot is looking at an electrode placement that was moved from two different locations. So very deep in position 2 and then pulled straight up to position 3 in that subplot. And then there are 9 individual neurons in different colors that are tracked during this movement. And at left and at right, 3 of those are called out with their different waveforms for different channels. And so during this forced or intentional removal of the electrode, we could track these signals moving along. And so here, this offers a potential for tracking neurons if, say, the electrode shifts, doing a much clearer analysis or identifying or sorting out individual neurons. And really, we think this could potentially mitigate against micromotion. So our prior experiments where we had bladder signals coming and going in an experiment, ideally here, we should be able to, as long as our software can track a cluster, this group of neurons, over time, we'd be able to follow them along. And then there's one other neural electrode array that we are collaborating or evaluating as possible use. And so this is a new Spark project. In this case, collaboration with Cindy Chestik and Pras Patel, where in their lab they've demonstrated that these carbon fiber electrodes, when implanted in the brain, can have very low tissue response. And so we're curious, can we do that in the periphery? So graduate student Alyssa Welly, along with John, and then Ahmad Juman and Elizabeth Batorf in our lab, are looking at developing a peripheral nerve application of this. So initially, we're sticking them in the vagus nerve, seeing if we can get single unit recordings from vagus nerve recordings, vagus nerve experiments, here in anesthetized rat experiments. And in some cases, we're seeing some nice large action potentials. We've also placed them in a couple of cat DRG, and we were excited to see that these sharpened carbon fibers went into dorsal ganglia, and in a couple of experiments we saw a nice bladder unit. So here in blue is an action potential from a neuron and had a very strong correlation to the bladder pressure. And so an array like this, where each fiber is 8 microns in size, so the same size or smaller than individual neurons, may mitigate against this tissue response. Of course, these are all acute experiments, and then the next question is can we put these arrays in place? Will they stay there? Can they offer long-term benefits? So in kind of summarizing, kind of reiterate that at dorsal ganglia and sacral roots, we can both monitor and modulate bladder function. And by using microelectrode array, we can get kind of unique signals into our system for decoding resolution. And by using a typical BMI algorithm with the Kalman filter, we can account for nonlinear relationships. But really, we need to see this scenario in sort of confounding non-bladder inputs, when an animal's moving around, when the tail's moving, when we're standing and walking, to get more of a robust bladder decoding algorithm. And so the closed loop approach shows potential for overactive bladder scenarios, but really if there's potential for translating this or continuing to study it, we really need better and more focused interfaces. Something with minimal invasiveness. There are approaches to access dorsal ganglia with a percutaneous approach. Some pain stimulation approaches are doing that may be translatable to applications like this. That's some of the future questions we're considering. So with that, I'd like to, again, acknowledge and thank the funding sources and also many members of the lab. So I highlighted some of the people who drove primary experiments, but many members of the lab have helped out during these overnight, multi-day sorts of experiments. And also acknowledge and thank collaborators on some of the work that was presented here. So with that, thank you for your attention.
All right, we can open it up for questions.
Nice talk, thanks. Just one question about the Utah array, the experiments you showed earlier. You stick that into the DRGs and mentioned that you were getting recordings from the axons. I thought you'd be recording from the cell bodies because they're producing a lot more larger signals.
Did I say axon specifically?
You did.
Did I? Okay. Well, it could be either.
Could be either. No, I agree. But
we don't know.
The cell bodies are so much stronger, I'm assuming. So.
Yeah. So it could be either axons or cell bodies. Odds are we're most likely recording from cell bodies. One thing we'd like to understand is which it is and whether we can tell that from the waveform that we see, because there are some waveforms that are non-traditional in their shape, and where you assume those are different parts of a cell body, or the glomerulus component where the stem axon is going into it, and others that might be more axon-like. The other component is when we're using these arrays, the tip location is more in the central part, and so there's probably more axons there, but I imagine a few cell bodies in the area. Maybe.
You mean the tips of it go to the central portion?
Yeah, when we hammer the arrays in, as opposed to being more of a superficial where most of the cell bodies are packed. But
So when you put the carbon fibers, the sharpened ones, do you aim for the surface of the ganglion or for the deep portion? Or?
Yeah, I mean, in that case, they were about 150 microns long, give or take, and so we're probably, somewhere around 50 to 100 microns into the cell, and so there we're getting maybe a third of the way into it, perhaps, in some of the smaller ones, and so that's.
So you should be right around where the cell bodies are, right?
Most likely.
And do you see bigger signals?
Well, they were big in these cases, but we haven't done enough experiments to kind of quantify that, but yeah.
Okay.
So yeah, thanks. Very interesting talk. I was wondering, how would the relationship between bladder pressure and volume and DRG firing change in a neurogenic state or in a pathologic state? Because these are mostly normal, intact experiments. Do you have any thoughts on that?
Yeah, so it's a good question. So there are two parts to that. I think we haven't closely looked at a relationship between volume and neurons, and so that's something where in most of our neurons we seem to respond more to pressure, as you saw some of the oscillations when they fired as the pressure was there. After certain neurogenic states like spinal cord injury, there can be a change in how neurons respond to changes in bladder pressure. So there's kind of two neurons coming from our bladder. There's the one that tells us, okay, the bladder's getting to be full, and in general, those maintain their relationships. I think that would be consistent. I think that's what we're seeing here. But there's also C fibers, or the pain fibers, coming from the bladder. So those are the ones that's kind of like, I really need to empty my bladder. After spinal cord injury, those become more active at low bladder volumes. And so those would be the ones we might be looking to record from. The challenge, though, is that they're C fibers, they're small fibers, and whether we're able to observe them is still be a question. Cool.
I was wondering if you could talk to, for the stimulation studies, did you see any relationships between how many locations you would stimulate and the effects you would get? Right, so for some of them you could get very, it looked like for some single locations you could get bladder excitation. Did you look at multiple sites or the effect of number of sites versus pressures generated or anything like that?
Yeah, so that's a question for, I wish we had infinite time and resources. There were a few cases where we tried to dial it down and stimulating 40 channels, 30 channels, and stepwise. And oftentimes it took, to get our best responses with 2 or 3 channels at the same time, which would match what we would see with many channels at once. But across those experiments, we didn't see any trends. It was always in one half or the other half of the ganglia necessarily. And so it was something that if we were to do that approach, we'd have to really map it out, which could take some time.
Well, maybe I should clarify then. So in your studies, were you stimulating all the sites you found that were excitatory?
It varied. It depended.
Oh, so
sometimes it was a few, other times it was more.
Okay.
That could vary.
So you mentioned a couple things that would suggest bladder state beyond pressure would be important, right? Like the nonlinear stress-strain curve of bladder filling, and there was another one. So I wonder, but your filters, like the Kalman filter, and they didn't seem to take that into account, right? So, the hysteresis was the other one, right? Of the response, right? So it would seem to me that they're not responsive to pressure, right? And the lack of Pacinian corpuscles in the bladder generally would suggest that from a biological perspective, that the sensory neurons are stretch sensors more than actual physical pressure sensors from a biological sign in the bladder. So I wonder if you can just improve the accuracy of your models, right, by taking some of the physics and biology into perspective, and then improve your closed loop stimulation.
Certainly there's a lot that we don't understand about how the signals go from changes in volume, changes in tension in the bladder wall, to what signals are being transmitted upwards. And a better understanding of that would definitely inform improved models. Yeah, I agree.
Yeah. And my understanding is that the sensory nerves in the bladder are likely responding either to stretch or tension rather than pressure directly. And there are mathematical correlations, if you know volume, between those. So anyway, something to think about.
Thanks. I was just in Toronto last week or so and talked to Paul Yu. Was he before you? I can't remember. Or after? It was after you, right?
He was before me.
Before you, really?
In Cleveland, he was before me.
Wow, okay. And he came up with this, after he left my lab, he saw the light, I guess, and worked on bladder.
Smart guy, yes.
And he came up with this thing, I don't know if you've seen it, on the saphenous nerve stimulation, a very small branch of, I think, pudendal nerve or some other nerve, I can't remember which one. Sural nerve, maybe a branch of the sural nerve. He says it's working really well as far as controlling. So can you comment on that, and how does that fit in with some of the stuff that you're doing?
So it's an interesting work that Paul's doing, and don't know the full extent, but it relates to the tibial nerve stimulation that I talked about earlier. So here people are getting that tibial nerve stimulation is once a week or multiple times a week at the ankle. And in that process, a needle is placed through the skin near the nerve itself. Well, the saphenous nerve, as you mentioned, the kind of distal branches go through the skin where that needle is placed. And so they've shown some studies, if you go more proximally, you get similar types of effects, so whether you're tibial nerve stimulation or saphenous nerve. Whether he's doing it in more of a continuous model, like some of the sacral neuromodulation, or periodic as tibial nerve stim, I'm not entirely sure. I think he might be looking to commercialize some of those aspects. But it's interesting, however, that there's leg muscles that are controlling our bladder function. I think.
Can you speculate on how that possibly works?
I mean, I think about it in some ways, I don't know, standing or walking around when we're running away from that bear and we want to activate our flight or fight response, we want to turn off our bladder, for example. It's an extreme example, but that's some of the thoughts. The other components are that these nerves, they share spinal levels with some of the pelvic nerves come in at, I mentioned S2 to S4. Tibial nerve is, I'm probably going to get it not entirely right, but they do innervate some of like S2 in humans and in higher levels. So there may be shared synapses in there. The interesting thing is it's been shown that a lot of the tibial nerve stimulation, you need an intact spinal cord, and there's different neurotransmitters that are active versus, say, the sacral nerve stimulation. So it's not the same paths, but there may be some overlap. And certainly there's a lot of work trying to understand tibial nerve, saphenous nerve, and what else may modulate bladder function and other pelvic organ function as well. Because a lot of this has off-target effects. Bowel function, sexual function are also modulated. It's kind of interesting. Very interesting. Everybody should try to work there. You want to work in the pelvic floor. It's more forget about the brain or the arm or the hand. That's where you want to be at. Right, Margot?
Absolutely.
All right.